Good evening, students. So this session will be dealing with the molecular structure and functions of nucleus and nucleolus. So to begin with, nucleus as a cell organelle was first described by or discovered by the scientist Robert Brown, as early as in 1831. Okay, so later the material of this, that is a nucleus, stained by the basic dyes. Okay, basic dyes, and it was given the name chromatin. Okay, by the scientist Fleming. Okay, so he said it as a chromatin. So in this electron microscopic picture, okay, so you can see the structure. Okay, as exactly it looks. Okay, so this is the nucleus, and surrounding this, that is endoplasmic reticulum. Okay, this is nucleus, nuclear membrane, and the darker central. Okay, so it is nucleolus, and the light stains. Okay, so which we are seeing, it is euchromatin, and darkly stained, that is a heterochromatin. Okay, and this is nuclear envelope. So as you all know, the euchromatin region where the active transcription you can see, and the picture is in a interphase nucleus. Okay, that means nucleus of a cell. So when it is not in a dividing phase, okay, when it is not in a dividing phase, so that's an interphase nucleus. So in the interphase nucleus, you can see these all things. Okay, so and it is a highly extended and elaborate nucleoprotein fibers. Okay, as I said, proteins, that's a chromatin, nuclear matrix, and one or more spherical bodies. Okay, nearly spherical bodies, that's the nucleoli, and singular it is called as nucleolus. Okay, so the cell nucleus is a membrane bond. Okay, so it is a membrane bond structure that contains the cell's hereditary information. Okay, and also controls the cell growth and reproduction. So, which is the main part of the cell, and the every information of life is there in the nucleus, and it is a command center. Okay, so it is important one command center of a eukaryotic cell. So it is commonly the most prominent organelle in a cell, accounting about the 10% of the cell volume. That is a 10% of cell volume is comprising the nucleus. Okay, so as prokaryote is a lack of nuclear membrane, so there you cannot see or you cannot differentiate the nucleus as nuclear membrane is absent. So there in that is in prokaryote, you can see a dispersed type of a DNA. Okay. So in general, the eukaryotic cell has only one nucleus. Okay, but in some extensions or exceptional case, some eukaryotic cells are anucleated cells. That is without nucleus, and that is example RBC. And in some are multinucleated. Okay, consists of two or more nucleus, slime molds, and also coming to the animals, sarcomere is a multinucleated cell, and also some of the epithelial cells also multinucleated cells. Okay, so as the nucleus regulates the integrity of gene expression, it is also referred as a control center. Okay, so it's a control center of a cell. So this is the three-dimensional structure of a nucleus. Okay, so it's surrounded by the endoplasmic reticulum cisternae, and these are the nuclear pores. Okay, and this is the nuclear envelope, so it is a double walled, and at the center you can see the nucleolus. Okay, so we are within this you can see even nucleoplasm. And this is the structure of a nucleus encompasses the mainly three parts, so four parts. Okay, so that is a nuclear membrane, nucleoplasm, chromosomes, and the nucleolus. Okay, so it comprises of these parts. That is nuclear envelope, nucleoplasm, nucleolus, okay, nuclear pore, chromatin. Then coming to the nuclear membrane. Okay, nuclear membrane is a double layered structure as you are seeing in a plasma membrane. No? So it is looking same and encloses the contents of nucleus. Okay, so within that you can see the nucleus. The outer layer of the membrane is connected to endoplasmic reticulum. Okay, so like the cell membrane, the nuclear envelope. Okay, nuclear envelope consists of a phospholipids bilayer. Okay, it is having a phospholipid bilayer. The envelope helps to maintain the shape of the nucleus and assist in regulating the flow of molecules. Okay, so in and out through a specialized structure called nuclear pores. Okay, so you can see the nuclear pores. Okay, within that the molecules, so they are transferring from in and out. Okay, such nuclear pores are the sites of exchange of large molecules, even the proteins and RNA between the nucleoplasm and also the cytoplasm. Okay, and within this perinuclear space, that is in between outer membrane and inner membrane, there is a perinuclear space and which is filled with the fluids. And then coming to nucleoplasm, so which is a cytoplasm of nucleus, it is a gelatinous substances. Okay, and it is also called as karyoplasm. And this is a semi-aqueous material, as we said, gelatinous, uh, to similar to the cytoplasm only. And mainly it is composed of the water and dissolved salts, enzymes, organic molecules, and they are all suspended within that. Okay, and the nucleolus and chromosomes. Okay, nucleolus and chromosomes are surrounded by nucleoplasm. Okay, so which functions to just as a cushioning. Okay, as a suspensions for the nucleolus. Nucleoplasm also supports the nucleus. Okay, it also supports the nucleus by helping to maintain its shape. Okay, and additionally, nucleoplasm provides a medium by which materials such as enzymes and also nucleotides, that is, it may be a DNA or RNA, can be transferred throughout the nucleus. Okay, substances are exchanged between cytoplasm and also nucleoplasm through the nuclear pores. Then nucleolus. So nucleolus is a centrally placed dense membraneless structure. It is a membraneless structure and it is mainly composed of RNA and proteins. Okay, and some of the eukaryotic organisms have a nucleus that contains up to nearly 4 nucleoli. Okay, some may contain, and the nucleus contains the nuclear organizers. Okay, and which parts of chromosomes with the genes and ribosome synthesis on them. And the nucleolus helps to synthesize ribosomes mainly by transcribing the assembling ribosomal RNA subunits. Okay, so in detail, nucleus we will be studying in next slides. Okay, these subunits join together to form a larger proteins, larger ribosomes, which are helping for the protein synthesis. And the nucleolus is disappearing. Okay, when the cell undergoes division and is reformed after the completion of a cell division. Okay, so that means at the time of cell division, the nucleolus is not appearing. And then chromosomes. These chromosomes, as you all knowing, okay, so these are the houses of chromosomes. Okay, nucleus is said as houses of chromosomes. Okay, okay, and chromosomes consists a DNA, so mainly DNA, which contains hereditary information and instructions for cell growth, development, and the reproduction. And the chromosomes are seeing as strings of DNA and histone. Because of histones, so they are also called as beads of string. Beads on a string also it can be called as. Okay, these histone proteins are some are octamers are also there. Okay, so when a cell is resting and that is not dividing, the chromosomes are organized into long enlarged structure called chromatin. Okay, so the when the cell is in interface stage, okay, so these appearance will not be seen. Okay, they are seen as a chromatins. Okay, so loosely arranged. And in the chromatin, two types of chromatins are there. One is heterochromatin and euchromatin. Okay, depending upon the functions. Okay, the heterochromatin is a transcriptly inactive form. Okay, where you cannot find a transcription phases and mostly present adjacent to the nuclear membrane. Okay, and euchromatin is a delicate and less condensed, and it is lightly stained. And this is majorly you can see the transcription phase. Okay, so apart from these things, okay, nucleus also contains some of the interesting things. Okay, that is a non-membrane delineated bodies. They are called as, and this includes causal bodies, gemini of coiled bodies, and a polymorphic interface karyosome association, that is pica, and also a promyelocytic leukemia, PML bodies, and paraspeckles and splicing speckles. Okay, these are all the things which are present in the nucleus. Okay, so these causal bodies, okay, they are uh, having a 0.3 to 1 micrometer in diameter, and they resembles just a balls of a threads. Okay, and a nucleus, each nucleus contains around 1 to 10 causal bodies. Okay, so they are coiling positive. Okay, they coil in a positive way. Okay, and these helping in the maturation of RNA processing. Okay, by maturing the snRNA, that is a small nuclear ribonuclear protein. Okay, so and also telomerase assembly also you can see, and also it helps for the modification of histone. And the Gemini of causal bodies. Okay, they are also same like these causal bodies. Okay, and they are helping for same biogenesis of this RNA, that is snRNA. They are called as gems. Okay, they are called as gems, and they are um, twins in a shape. Okay, they look like a twin shapes, and they contains same snRNA, that is a small nuclear ribonuclear protein. Okay, and they are coiling negative. Okay, they coil negative, and they are assisting the causal bodies. Okay, so in a biogenesis of a RNA. And then pica bodies, that is a myelocytic leukemia. Okay, they are helping mainly transcription silencing complexes. Okay, so they but the functions of this pica is unclear, but especially they are associated with the chromatin. And next is the PML bodies. Okay, PML bodies, they are called as nuclear domain 10, that is a ND10. Okay, so they are helping for the subnuclear structuring, and these are spherical, and the number of these bodies are 10 to 30s, and it's having a diameter of 0.3 to 1 microns. Okay, and also they contains some proteins. Okay, leukemic proteins, and they are suppressing the growth and transformation after formation of a new cell. Okay, they are controlling the cell growth. And paraspeckles, another type of bodies. Okay, they are irregular shaped subnuclear structures. Okay, they connect to interchromatin space. Okay, and they are placed in interchromatin spaces of a nucleus, and the shape of diameter is 0.2 to 1 micron. Okay, and the another type of speckles are there, that is splicing speckles. they are splicing the snRNP. snRNP means that is a small nuclear ribonucleoprotein. Splicing means it is a scissoring. And another set of spectral fibrils are there, perichromatin fibrils. Okay, they are subnuclear fibrils and they are rich in RNA. They are also helping in a splicing of free mRNA and also polyadenylation. That is the addition of the adding one nucleotides. Okay. So then functions of nucleus includes mainly they are uh, important as we are seeing. Okay, so they are like centrally, and the main function of the cell nucleus is to control gene expression, mainly gene expression, and mediate the replication of DNA during the cell cycle. Okay, it also controls the hereditary characters and also it transforms the hereditary characters. Okay, they are helping for the DNA replication. Okay, and also transcription and the protein synthesis. Then they also stores the proteins. Okay, so and in as in a nucleolus. So during the cell division, chromatins are arranged into chromosomes. Okay, so loosely arranged chromatin threads are forming a chromosome structure during the cell division, and division takes place. And they are helping for the protein synthesis as in ribosomes, and selective transportation of regulatory factors and energy molecules through the nuclear pores. If any important proteins are synthesizing in the nucleus, okay, so they are transferred from nucleoplasm to cytoplasm with the help of nuclear pores. So this is the structure where you are seeing the euchromatin, so where it is a lightly stained, and the heterochromatin where it is a darkly stained, and this is nucleolus. Okay, these are the fibrils, granules. Okay, and these are nuclear organizing center within a nucleolus. Okay, so you can see the exact structure. Okay, but these all chromatins when in a cell division they undergo formation of chromosomes. Okay, chromosomes as you are seeing a shapes, that X like shape. Okay, so then coming to nucleolus. So nucleolus is the distinct structure present in the nucleus. Okay, so distinct structure present in nucleus of eukaryotic cells. Okay, primarily, so its work is to assembling the ribosomes and alteration of transfer of RNA, the tRNA, and also sensing cellular stress when in a cell under stress. Okay, so it will decide whether the cell is to death or to repair. Okay, that's all thing done by this nucleolus. Okay, and it is composed of RNA and proteins. Okay, which form around specific chromosomal regions. Okay, it is the main components of nucleus, important part of nucleus. The chain of RNA and DNA along with other components form the structural components. Okay, so structural components are formed by RNA and DNA. The main components of nucleus, okay, that is ribonucleic acid, deoxyribonucleic acid, and proteins. And structure, you can see uh, it is having a well-ordered structure with the four main ultra-structural components. Okay, so main four ultra-structural components. Okay, you can see in this diagram. The one is the fibrillar centers. Fibrillar centers. You can see here ultra-structure fibrillar centers. Okay, so it is the place where the ribosomal proteins are formed. Okay, ribosomal proteins are forming, and when the protein synthesis is happening, then these all coming together and to form our ribosomes of a two subunits. Okay, and then granular components. Okay, these are the granular components. Okay, so they are pre-ribosomal assembling. Okay, before ribosomes are formed, these components have a rRNA, that is a ribosomal RNA that binds to ribosomal proteins, then forming the subunits. And then dense fibrillar. Okay, these are the pre-RNA processing. Okay, dense fibrillar components are there. So So it has a new transcribed RNA. After transcription, the new transcribed RNA will be there, and which connects to the ribosomal proteins. And then nuclear vacuoles are there. Okay, nuclear vacuoles. So they are present only in plant cells. The ultrastructure of nucleolus can be easily visualized through the electron microscope only. The arrangement of the nucleolus within the cell can be clearly studied by the technique. There is a fluorescent recovery. Okay, so it can be seen under fluorescent microscope. Okay, so this is the structure where you can see, and the nucleolus of a plant species. Okay, so this is the nucleolus of a plant, and this is the animal cell. Okay, so plant cell nucleolus is containing a higher amount of ferrous ions. That is iron content. So it is looking much darker than the nucleolus of an animal cell. Okay, you can compare with these cells. This is plant cell, this is an animal cell. Okay, so the nucleolus here and nucleolus here, you can have a differentiation among them. Okay, so then a nucleolus functions mainly the 25% of the nucleus is composed of a nucleolus. Okay, so primarily it is having a function as a ribosomes productions in it. Okay, so this is the way of ribosomes producing. That is it is called as ribosome factory. And also another next will be the control of a cell cycle. Okay, so it also controls the cell cycle and cell proliferation, and it also helping in a stress response. Okay, that is the stress sensing and tumor surveillance pathway. Okay, so that means the control of a cell growth, and also if the cell is under stress. Okay, when it is under stress of any foreign particles, okay, or foreign impulses, then it will decide whether the cell is to be repair or cell is to be death. That is a programmed cell death also done by this nucleolus. You can see the regulation of protein stability and also apoptosis. Apoptosis is nothing but a programmed cell death. That is a suicide of a cell. And also it helps when the virus enters into the cell. Okay, so it also try to destroy the virus life cycle. Okay, and also it helping for the telomere metabolism. Telomere means it's the end of chromosomes. Okay, end part of chromosomes, it is also metabolized by these nucleolus. And also as I said, so it will helping in a DNA damage or DNA repairing mechanism. And also it helps for the maturation of small RNAs. Okay, small nuclear RNAs are also ribonuclear proteins also matured with the help of nucleolus. Okay, then what is the difference between the nucleus and nucleolus means? Nucleus is large in size, nucleolus is very small in size, and nucleus is bound by nuclear envelope. There is a membrane bond, but nucleolus has no limiting membrane. It is not surrounded by any membrane. Okay, and also nucleus contains a chromosomes, but it does not hold any chromosomes. Nucleus will not be holding any chromosomes. Okay, nucleus is rich in DNA and genetic materials, but nucleus is very much rich in RNA and also the proteins. Okay, that is a ribosomal proteins. Okay, so this is all about the nucleus and nucleolus. Thank you.